Okay.
Uh, it's such a pleasure to invite um, Sean Crosson, um, a super all-round physicist, biologist, whom I've known for a long time, to help us launch into anniversary edition Living Histories in 2024 November. Without further ado, Sean, please tell us about all these many living histories, stories that you could possibly tell us.
All right. Well, thanks for the introduction, Sri, and thanks. Yeah, just thanks for the invitation. It's really an honor to be here. Y'all can hear me okay? All right, cool.
Yes.
Yeah, my plan was to just, I looked at a few of these and just provide you with some view of my path to how I ended up doing what I'm doing now. I'm going to share my screen and just show you some slides. So, all right. Yeah, I grew up in Texas, and this is a view of where I grew up. It's Grayson County, Texas. The nearest big city is Dallas, but this is just my brother actually saw this map and he didn't quite like the view, but I'm showing it anyway, just to give you some perspective on where it sits with respect to the Red River, which is the Texas-Oklahoma border. So it's a county that borders Oklahoma. This is a rural area in northeast Texas. And this is just to show you, this is the view out the back of the place. And this is wheat. We don't farm, but it's surrounded by plants and animals. That's my youngest daughter out in the field searching for cell signal. And this is the view out of the front. There's a bunch of cows. And so that's just, I think that growing up in this rural area, I really think shaped how I see biology, and I really moved in a more agricultural direction in recent years. But this is just to sort of show you where I'm from. And, it was, I didn't come from a sciencey family, really, not at all. My dad installed window coverings and built custom window coverings. And my mom worked in an interior design store. They drove toward the city to work, but I'm the oldest of three, and my little brother there is on the left, and my sister's on the right. This is us now. This is the same. You can see the wheat field in the back, but we're all grown, and I'm the only one who's not there within that little map I showed you. I'm the only person who doesn't live there anymore. So my brother and sister, their kids are all still back in Grayson County, and I do miss them, and I like to get back there. So that's just to sort of show you where I'm from. Um, I mean, I went to it was a rural high school, and there was 47 kids in my class, so I was one of 47, and certainly students there, did go to college. It wasn't necessarily the most common path at that time, but I was actually recruited. I had this idea that I wanted to go to college, and I knew that I liked biology. I knew that I liked science as a kid, but I wasn't really sure what it meant to do research. And I think if you like science, then maybe you would be a doctor or a veterinarian. And I think so. For the longest time, I thought that's what I was going to do. And I was actually recruited to go to this little tiny college. It's less than 1,000 students, Earlham College. So I left Texas and I went to Indiana. I went to Eastern Indiana, and I was recruited there to run track, cross country and track and field. And by the coach there at the time, Pat Thomas, who I owe him a lot. If it weren't for Pat, it weren't for this track and cross country coach calling me, I wouldn't have, I wouldn't be where I am now. So I think Earlham has really, shaped my view of the world. It's a small Quaker school. And it's just a lovely place. And I'm super happy to say that my youngest kid is a student there now, is actually an undergrad there. And when I graduated from Earlham, I thought that I was going to go to medical school, and I ended up applying to medical school. In this year, I did AmeriCorps for a year, which is a national service program, and I was in Austin, Texas, and working for a while in a shelter, a homeless shelter, and then ended up working for the State Coalition for the Homeless, and that was an interesting view into the nonprofit sector, just to sort of see what that world is like. But I think during that year, particularly working in the shelter, I realized that maybe I'm not a caregiver. And I don't mean that like I'm a I don't see myself as a bad person, but I think to give care to people requires a special kind of person. And and I think it was this year that really convinced me that maybe I should maybe just follow my nose and my interest in science and research, right? So I ended up backing out of, I got into UT Southwestern medical school and but I ended up backing out and following my girlfriend at the time, now my wife, on graduate school interviews. And we ended up choosing a PhD program at the University of Chicago. And so I was in the biochemistry and molecular biophysics program. And I have to say my first year there, I didn't like it at all. Actually, I didn't like graduate school. Um, and I, for a lot of reasons, I think, I was at this teeny little college and it was a super nurturing place and the quality of teaching there was outstanding. And certainly I had great teachers at the University of Chicago, but it was just, it's a different kind of game at a place like Chicago. And I almost quit graduate school, but um, I didn't. And I hung in there and I found a mentor, Keith Moffat, who's a biophysicist and structural biologist. And in Keith's lab, I was able to sort of grab onto some first principles things, and that was a really fantastic lab for me. And the community there was really great. Community, I think, as many of you can attest, is super important. And this was the first project that I was involved in. I wasn't first author in this, but I am an author on this paper. It's involved in using. It's about using polychromatic X-rays to do time-resolved X-ray diffraction experiments. So it was really in the method development phase of time-resolved X-ray crystallography. And so I did my PhD in Keith's lab and developed a new system working on protein photoreceptors. Ultimately, we were trying to get it to the point where we could do time-resolved X-ray diffraction, and we never got there. But I, I was a biologist. I was a biology major at Earlham, and I wanted to get back to biology, and I was always interested in bacteria. And so at Stanford, I joined the group of Lucy Shapiro, who of course is a legend in microbiology. And this was another really just fantastic community. There were so many great people in Lucy's lab at the time. It really was like, the all-star team. And I learned so much. It was such a fantastic place to be. And I really became a microbiologist there, but then went back to Chicago, and I actually went back to my old department, but I was really working in a very different field, after just a few short years at Stanford. And I'm super grateful that they had me back there. And I think Chicago is always going to be my intellectual home. And I was there for 14 years on the faculty. But, I think I'm kind of a restless person. And what I think is cool changes. And I think for better or worse, my group jumps around a lot. And I was really starting to get interest in agricultural research and more applied research, and had the opportunity to move to Michigan State University, which is one of the United States' land grant colleges, right? And so I think that the mission is just completely different here. And I think culturally it's very different too. I mean, you can see like the giant football stadium in the background, right? So it's a large state university, land grant institution, but it's a really fantastic place. And this is where we currently are, to just think more about agriculture, think more about application, and taking and bringing the fundamental work that we do to some sort of application is really what we're trying to do here. And I just wanted to give you a brief view of what we're doing, just some current projects. And as always, like these are projects where we have a really great opportunity to collaborate and work with people. And I've collaborated with you, I've collaborated with Sri, right? And I think these are the most fun projects where you can talk to people and hear other people's ideas, and it can really move your science in new directions. And for me, that sort of helps fill this restless void that I feel sometimes as a scientist. So we've worked for Caulibacter for many years, and it's a fantastic model system. But now we know that Caulibacter lives in the soil and is associated with plant roots. And now there's good evidence that this microbe can help support crop growth and soybean growth, actually. And so, we're trying to now, how can we leverage what we're learning from a basic perspective to maybe improve crop resilience and crop productivity, which I think this is going to be as we look at increasing shifts and extreme changes in our climate. I think this type of work is going to be really important for sort of stabilizing food production going forward. And then we're also thinking about microbes and interacting with the host environment. There's of course microbes that live in our gut, and this is we still have collaborations with Qin Cheng's group and Mitch Sogan and Murat Aaron, thinking about how microbes might impact inflammation in human hosts. And we're also working on this microbe that's associated with many animals, right? It's associated with, we work on Brucella abortus, which you find in cows and causes, disease in cattle, and it's a zoonotic pathogen, can cause disease in humans. But it's an incredibly diverse, the animal host that these microbes infect, Brucella, the genus Brucella, it infects an incredibly diverse range of animals. And so this has been really fun, ongoing collaboration with the USDA and with veterinarians here, trying to think about, how these microbes might impact animal health, but also human health. And so I think that's where we stand now. And I just, as always, I sit here and mostly write. I'm still doing some experiments, but it's the group, it's the people who we collaborate with who really make this career such a joy. And it's just, so thanks for the opportunity to sort of tell you about my path and where I'm coming from. Thank you. That's all I have.
Thank you, Sean. On behalf of the audience, I'm clapping. Thank you so much for such an inspiring talk. There's been a few questions on the same theme, so I'm going to combine and ask, which is, you described so poignantly how you were raised, the surroundings in which you were raised. Would you please connect the dots from there to your current research interests? In particular, what drew you to microbes?
Yeah. I think sometimes you just end up in space by chance. And I was working on plant photoreceptors in Keith Moffat's group, really doing, very atomic level work, trying to understand how photon absorption impacts conformational change really at the angstrom scale. And I found a paper by Lucy Shapiro that showed that there's certain proteins in a bacterial cell, which is very, very small, will localize to particular positions. And that idea, that notion captured me, just that proteins can get addressed in these teeny little cells. And so I contacted Lucy. And then I went to Lucy's lab, and now I work on microbes. And so if I had ended up postdocing in another lab, like I interviewed in a physical organic chemistry lab, and I interviewed in a nuclear pore complex lab, if I had gone to those places, maybe I would be doing something else now. So I don't think there was ever like some sort of line in my mind, 20 or 30 years ago that I was going to work on microbes. I think it's just, I think it's serendipity. It's just kind of, I think that's how life is, or that's how I see it at least.
Music to our ears
Yeah.
of the series. I'll round it off with one more question, which is that so such few members of faculty in our experiences come from first-gen backgrounds. Would you please speak to the unique challenges and learning opportunities you had coming from the background you did, which is unusual for somebody who is now a full professor in a US university?
Yeah, I think it's not something that I thought a lot about growing up. I always felt very supported, by my parents. I do think that what was hard, and then I went to this small school in Indiana where there were, other kids like me there, actually. Now, that's one of the things I love about Earlham College, is it's an incredibly diverse place, both racially, ethnically, but socioeconomically. And then when I went to the University of Chicago, maybe that was one of the things that was so hard for me is I felt this way, and my wife felt this way too. There was just some people there who like already understood the game, and we just felt very mature and much more sophisticated than I did. And so I think that that was just that was the hardest thing for me to overcome in graduate school is just to sort of I felt like I particularly my first year that I just didn't belong or I just didn't have the game or I didn't, I felt like people were just so far ahead of me in their thinking. Um, so and I don't know how you overcome that other than if you're around people who trust you and support you and tell you that it's going to be okay and just keep throwing punches, and you'll get there. To have people believe in you, I think, is the most important thing. If you don't have that, then you really are at risk then of just dropping out, which I almost did. I don't know. I hope that answers the question. I don't know if there's a good answer to that.
Thank you for sharing that. Yes, it did. And thanks again for a very inspiring talk and for these thought-provoking answers. In the interest of time, I'm closing the recording.